Hello everyone, my name is Lemu. I'm a second-year medical student at the Brody School of Medicine. Today I'll be presenting to you all a paper looking at the effects of bee venom on the pro-inflammatory responses in a specific cell line of mouse macrophages. Serious inflammation can lead to an array of diseases, including septic shock, rheumatoid arthritis, and various cardiovascular diseases. Excess production of nitric oxide and prostaglandin play an important role in inflammation. In order to suppress inflammation, excess nitric oxide and prostaglandin have to be addressed. Bee venom has been known in traditional Oriental medicine to relieve pain and treat inflammatory diseases. However, the exact mechanism of action behind the anti-inflammatory property of bee venom is not fully discovered. The authors sought out to understand this molecular mechanism by looking at the effect of bee venom on excess production of nitric oxide and prostaglandin. Inducible nitric oxide synthase and COX-2 have been studied as the basis for excess nitric oxide and prostaglandin, respectively. Furthermore, it was important in this study to understand how transcriptional factors such as NF-kappa beta play an important role in the expression of inducible nitric oxide synthase and COX-2. In order to conduct this study, cells from these mouse macrophages were cultured at 37 degrees Celsius with antibiotics and mediums suitable for these cells. The bee venom was obtained from the bee sac, diluted with water to make a solution. This solution was then sterilized while maintaining the major components of bee venom. In order to measure the nitric oxide production, nitrate levels were used as a proxy. This measurement for nitric oxide production was following a 2-hour lipopolysaccharide or LPS stimulation and sodium nitroprusside or SNP stimulation. The cells then underwent a 5-hour treatment with bee venom. RT-PCR was also performed using primers for specific inflammatory markers, including NF-kappa beta. The results were interesting. If we look at Figure A, we see a dose-dependent reduction in the nitric oxide production with bee venom treatment after cells were stimulated with LPS. Similar pattern is observed when stimulated with SNP, but a much less reduction. Cells labeled here as normal were those not stimulated with LPS. The control here are cells that were stimulated but did not receive bee venom. If we look at Figure B, the RT-PCR results showed that compared to the control, which again is LPS only, expression of these inflammatory markers was reduced. Not a dose-dependent pattern as seen with the nitric oxide formation, but a marked decrease with bee venom is observed. Beta-actin was used here again as an internal control. In conclusion, bee venom has anti-inflammatory activity that decreases LPS-induced nitric oxide formation. The inhibition of nitric oxide formation was attributed in the study to be due to the inhibition of inducible nitric oxide synthase and COX-2 expression through the suppression of NF-kappa beta and MAPK activation. Furthermore, the effect of bee venom on NF-kappa beta is through the inhibition of its dissociation. NF-kappa beta is normally inhibited by binding to a known inhibitor, I-kappa beta. By inhibiting this dissociation, NF-kappa beta is not released to induce expression of inflammatory enzymes, including induced nitric oxide synthase. Although the authors have suggested bee venom treatment can be applied to inflammatory diseases, a limitation is noted in that anti-inflammatory component of bee venom has not been identified. Thus, the authors could not rule out scavenger activity. However, other studies have noted the anti-inflammatory effect of bee venom outside of scavenger or toxic effects. Thank you for listening.